Hi, Jay.
Hey, how are you?
Great. Thanks so much for joining us today.
Sure.
Yeah,
No, I'm happy to do it.
yeah, we're delighted to have you on board.
Let's make sure the screen share thing is going to work. Do you see that okay?
Yep. Yeah, looks good.
Hey, Jay.
Yep.
Eric and I adjusted a few of our questions for part of the webinar today. Would it be okay if we massage those a little bit to be more in line with your presentation?
Yeah, I saw those. That's just fine.
Sounds great. Thanks.
So in the screen share, let me go back to that again. Can you just see the slides, or can you see all the slides on the left-hand side too?
I can see the slides on the left hand too. If you go to the presentation mode, that should stop. There we go. Now I only see the slides and your face.
Okay.
Greetings, everyone. Welcome to the fifth and final part of our new webinar series in partnership with the Montana Natural History Center and the University of Montana, The Age of the Coronapocene. I'm Jennifer Robinson, Education Director for the Montana Natural History Center and host of this webinar series. Our co-host today will again be Drew Lefebvre, the Montana Natural History Center Museum Programs Coordinator. She will be working in the background and available to answer questions and address comments using the chat box. Please let her know if you have any technical difficulties or questions during the webinar. We will also be streaming this webinar live on Facebook as well. So if you have any questions during the Facebook stream, you can also leave comments in the chat feed through Facebook. This webinar will again be supported by closed caption capabilities. The wonderful Caroline Habish will be working in the background to provide text for our speech if you need assistance in following along with the program. Please click on the link in the chat box. Drew will leave that there for you. This will open a separate window where you will be able to read what is being said on the screen as we are speaking. Please note that the view cap for the closed caption is only 50 individuals. Please only use this assistance if you need it, so we have enough space for all of those who require closed caption assistance. Throughout the webinar, we will repost this link into the chat box and through the Facebook Live chat feed for those who join late. Back with us today are members of our expert panel of scientists and educators from the University of Montana, and they're ready and excited to start more discussions about the science behind the novel coronavirus COVID-19. As a reminder, our panel are not medical doctors and will not attempt to try to diagnose symptoms in this program. Later in the program, the audience will have an opportunity to ask our panel questions about the virus and living with COVID-19, but note that we will not be taking questions seeking medical advice. Please seek your own medical help if you have questions or concerns about your health. With us today, we have a new expert guest on our panel, Dr. Jay Evans. Jay is a research professor in the Division of Biological Sciences and director of the Center for Translational Medicine at the University of Montana. He has worked in the biotechnology and pharmaceutical industry for 15 years at GlaxoSmithKline Vaccines, where he held various positions, including senior scientist, investigator, and project leader. While at GlaxoSmithKline Vaccines, Dr. Evans worked on the discovery and development of novel vaccines, adjuvants, and immunotherapeutics. Dr. Evans has also worked on HIV immunology, stem cell transplantation, and lentiviral-based gene therapy systems. Today's discussion will focus on Dr. Jay Evans' expertise on the science behind how vaccines work and how that relates to the work being done today to combat and develop a vaccine for the coronavirus. During the question and answer portion of the webinar, we will be joined again by by Professor Erick Greene and Dr. Rosalind Lapierre. Today's webinar will focus on the science behind how vaccines work and the processes in place in developing one for COVID-19. Dr. Jay Evans will start us off with a brief introduction about the COVID-19 vaccine program from the UN Center for Translational Medicine, and will transition into an interview-style discussion on topics related to vaccines and COVID-19. During the Q&A, you will be able to ask the panelists questions and have them answered live. To ask questions, you can use the chat box or the Q&A feature of Zoom. You can also leave questions in the Facebook live stream chat feed. To let us know of any technical difficulties or to communicate with the co-host, please use the chat box. Let's go ahead and try and use that feature now to go ahead and type into that chat box and tell us where you are all tuning in from. Looks like we've got some viewers from Missoula. Great. Moab, Utah, Frenchtown, awesome. Folks from the lab at the university, wonderful. More from Missoula, that's great. Thank you so much for telling us where you're tuning in from. Lolo, great. Okay, let's go ahead and jump in and get started. I'm gonna turn it over to Dr. Jay Evans. Can you start us off and tell us more about the COVID-19 vaccine program you've been working in?
Absolutely. I'd like to start by, of course, thanking the organizers for putting on this great series. We're all, some of us are quarantined at the university, some at home over the last few months, so it's great to have access to relevant information as to why we're all there and why we're stuck in the places some of us are stuck in. So the university has a kind of a fairly new program in the Center for Translational Medicine. We'll talk a little bit about what the Center for Translational Medicine is, kind of where we came from, why we're here, and finally the topic we're all here today to learn about more is the coronavirus. University of Montana has an active vaccine program on coronavirus ongoing right now that we hope will move towards human clinical trials here in the next 18 to 24 months. That we're pretty excited about, and we'll talk a little bit about that today. Let's see if my slide changer will work. There we go. So just briefly, what I'm going to talk a little bit about today is just an introduction as to who we are, what is the Center for Translational Medicine at the University of Montana, what do we work on, a little bit about our vaccine research and development team, and then a few slides about our specific approach to developing a coronavirus vaccine. So first off, what is translational medicine? It's a topic that covers a very broad range areas, everything from the early basic discovery research that people think about happening in a university setting, all the way up through the drug development discovery pipelines into phase 1, phase 2, and phase 3 clinical trials, till finally we have an approved medicine. This is a very long process. For most drugs, this can take 10 to 15 years. So we get a lot of questions lately about, well, how are you going to change a 10 to 15 year process to an 18-month process you've heard about for the coronavirus vaccine? And it is a challenge, but it is possible. As you can see, it's also very costly. You hear big numbers being thrown out by Congress and NIH about what the monies they're putting into development of a coronavirus vaccine. And these are real numbers. I know one company that I was on a call with a couple weeks ago that received nearly $500 million in their coronavirus vaccine program. As you can see, these programs often take anywhere from 800 million to up to $2 billion to develop a vaccine from the discovery stage all the way through an approved clinical product. So what is our focus area at the Center for Translational Medicine at U of M? We really focus on taking discoveries that happen in the labs at the University of Montana or MSU or other places in the state and helping investigators move those towards a phase 1 clinical trial. There's kind of a gap in knowledge that exists in a lot of university settings. And the goal of our group is to help people bridge this gap and get those discoveries to places where they can help people. Most of our group worked in industry for a lot of their careers, and so we focused in this space. And so partnering with the University of Montana and starting the Center for Translational Medicine, which we did in 2016, has been a worthwhile adventure, a good thing for Montana, and really a good thing for our group and hopefully the nation as well. So the Center for Translational Medicine at the University of Montana has a core group that works on vaccine research and development. We started at University of Montana in 2016 with about 15 people that came up here from GlaxoSmithKline down in Hamilton. Since then, we've grown to a team of more than 40 that's specifically focused on vaccines and immunotherapy. We do everything from initial vaccine design and discovery up through animal models and safety assessment that has to be done with any new vaccine, making it through that product development process, and ultimately to phase 1 clinical trials. You can see there a list of active vaccine research programs that are currently funded on our team at University of Montana. The top one there is, of course, the COVID-19 or SARS-CoV-2 project, which we started in February. We have a strong research program on influenza viruses and influenza vaccines, tuberculosis, Pseudomonas, E. coli, pertussis, better known as whooping cough, Lyme disease. We have oncology vaccine and immunotherapy programs. We have programs in allergy, and lately you might have, if you've been following the news, you also know that we're working on an opioid vaccine program, which is a kind of a novel target to develop a vaccine against. We bring together groups in chemistry, formulations and analytical sciences, as well as immunology, all together in one team at the University of Montana to move these candidate vaccines from discovery up to human clinical trials. We've only been here since 2016, but we are really a multinational team. This just shows the map of some of our collaborators just in the US. So the University of Montana is really at the center of vaccine discovery and development with a lot of partners throughout the country, and we can't do this alone. Our coronavirus response includes a group of investigators at Boston Children's Hospital, as well as at Mount Sinai, Icahn School of Medicine at Mount Sinai in New York. And together, these teams are working towards a solution to different vaccine platforms. And each of the people listed on there are partners with us in one of these vaccine platforms that I mentioned on the previous slide. So it's exciting to be part of a team like this and also go to live in a place like Montana. So a little bit just about vaccines in general. Anybody who has kids knows the number of vaccines that are available. It's kind of scary at times, and it's actually generated even some anti-vaccine movements that have been cropping up and actually creating some issues and with some kids and outbreaks of things that were once thought fully preventable. Just some examples here of a couple of really successful vaccines on the list. You can see measles in the bottom and polio. When these vaccines became broadly used, you can see the reduction in measles cases and polio cases dramatically dropping off with the development in of these vaccines. Disease like polio, you never hear any about anymore. And that's largely due to the vaccine efforts. So, you know, vaccines are really a fundamental driver for improvement in global health. They're one of the most safest and most cost-effective medical advances in healthcare history, second only to clean water. Each year, vaccines prevent up to 2 to 3 million deaths, and 750,000 children are safe from disabilities. And just in 2016, it's a little bit old, the date's a little old, over 116 million doses of the DTP vaccine were given to children. But despite all of these advances, about 1.5 million children still die annually from vaccine preventable diseases. So we can do better, not only for existing things that we have vaccines against, but also new and emerging diseases such as the coronavirus. So how's the vaccine industry changed over the years, and what's doing to help develop a next generation of vaccine? Most of the early vaccines that were developed 20, 30, 40 years ago were based on non-replicating, inactivated either viruses or bacteria. That is, they take a whole bacteria, they take a whole virus, they inactivate it, and then they use that as the vaccine. They were very effective, highly immunogenic, but as you see, the tolerability bar there is very small. They weren't very well tolerated. You had a lot of adverse events. You had a lot of side effects to those. So over time, vaccines have gotten safer by using subunit vaccines or part of viruses or bacteria to make those vaccines. And more recently, we're using what we call purified antigens. That means we take pieces of the virus or pieces of the bacteria, we purify them to really high levels of purity, and then we use that as the vaccine to give to people. They're extremely safe, but they also lack the immunogenicity. They don't drive the immune response that the whole virus or bacteria do. So what we have done here at University of Montana, and in our work we did down at GSK in Hamilton, is we add what's called an adjuvant or a delivery system to this purified antigen. And that is we take components of the virus or bacteria that help stimulate the immune response, and we add those back to the vaccine in a safe way that keeps it so they're very safe, but also increases the immune response to give broader protection and longer lasting protection. And so that's exactly what we're doing here at University of Montana for the current SARS-CoV-2 vaccine project. Again, like I said earlier, this project is in collaboration with investigators at Inimune, which is a company, a biotech company right here in Missoula, as well as Icahn School of Medicine at Mount Sinai and Boston Children's Hospital. We started these investigations earlier this year. We started discussions in January and actually started the lab work in February of this year. The vaccine adjuvant and deliver those immunostimulants and delivery technologies we're using were discovered right here at the University of Montana or Inimune, and these are the ones that are being tested in the facility today. We currently have ongoing animal studies, so we have lead vaccines are currently being tested in animals here at the University of Montana. We also have challenge studies going on down at Rocky Mountain Labs in Hamilton. Our vaccine is based on the major, what we call the surface proteins of this virus, oftentimes referred to as the spike protein or the S protein. This protein is being made both in New York at Mount Sinai as well as right here at University of Montana. And the goal is to generate an antibody response against that protein, which will help protect people that will get infected with the virus. And I'll explain a little bit more about that here in a minute. The ongoing studies right now are to optimize the best vaccine. That is combining the right pieces of the virus with the immunostimulants you need to add to boost the immune response and the right way to deliver it. Once we have that correct combination, we'll test those in animal challenge models where they're developing models now where you can actually challenge some specific strains of mice or potentially hamsters or ferrets with the virus. And this to do two things. One is to show that it's effective, but also to show the safety profile. Because with this rapidly as these vaccines are being developed and put into people, you want to ensure that they're always safe. The last thing you want to do is make somebody worse. And then hopefully that data will be used to support further development of those vaccines towards human clinical trials. So a little bit about our target that we're focusing on at As I said here on the left-hand side of the slide, you can see a schematic of the virus. That red protein that sticks out, we call that the spike protein. That's what binds the receptor called ACE2 on human cells, especially in the airway epithelium, and then allows entry of the virus into the cells and start the process. So if we can generate a vaccine using that spike protein, and antibodies against that spike protein could then bind to the virus and prevent it from infecting cells. So we have two different targets. One is the actual receptor binding domain, which we call the RBD domain, which we target antibodies against, and we're also using the full-length spike protein for the vaccine studies at University of Montana. And so how are we going about this? Our technology at University of Montana is focused on, like I said earlier, immunostimulants, formulation, and delivery approaches. Other groups around the country are also targeting the spike protein because it's the natural place you would look for a vaccine. But what's unique about our approach here is the unique combination of immunostimulants and delivery methods that we have. So if you can package, as an example of a vaccine delivery platform is shown there on the left, if you can package the spike protein in a way that presents it in the right way to the immune cells and stimulates those immune cells with an adjuvant, you can have an effective vaccine that's showed on the right, which will enhance the immune response. You can have a broader and more durable, long-lasting response, immune memory, which we'll talk about just a little bit. And this all happens through vaccine formulation and the technologies that we're doing in-house here at the University of Montana. Interestingly, a second approach that we're developing, and this is actually being done by a company here in Missoula called Inimmune. There's a study going on just this week down at Rocky Mountain Labs in Hamilton to evaluate. We have some of these immunostimulants are such strong immunostimulants, they boost your immune system in a way that helps protect you from a viral infection. It's not a vaccine, it's more of a drug because it stimulates your innate immune response and allows the body to respond faster to the virus. So a single intranasal administration of one of these immunostimulants we've shown can protect against respiratory infections, including influenza, RSV, and several other viral or bacterial infections. We haven't shown that this works against coronavirus, but we expect it will, and we should have those results within about 10 days, which is very exciting because it could provide us another tool to use not only against this virus, but against other emerging viruses that are acquired via the respiratory route, and maybe help us gap the time it might take for a vaccine to be developed. It's one last slide kind of about future vaccine research at the University of Montana. We've only been here since 2016, but Missoula and the community has been very welcoming. The University of Montana has been a great home for us, and we've developed a world-class vaccine center right here in Missoula. Since 2016, the center has generated about $45 million of new vaccine research funding for the University of Montana and the state of Montana. And hopefully several of the vaccine candidates and technologies that we've been developing over the last 4 years will be in human clinical trials in the next 18 to 24 months. And we hope one of those will also be a coronavirus vaccine candidate. With that said, there's great opportunities for both undergraduate and graduate students to participate in this research. All of our labs have students in them working on projects that can have a great impact on human health. And so that's kind of a brief overview of our project and what we're doing. I think we'll probably open it up to some discussions and some questions that people have about everything from the vaccine to coronaviruses in general, or even other vaccine programs we have ongoing at the university.
Great. Thank you so much, Dr. Evans, for your introduction to the COVID-19 vaccine program. I'd like to follow up with some more questions we have for you about the science behind how vaccines work in general. And Professor Erick Greene is also going to be asking some questions with me as well. So here's our first one that we have. In your presentation and around, we've been hearing a lot about antigens, adjuvants, and antibodies, and this gets confusing some of the time. Can you help us understand those differences?
Yes, absolutely. So the antigen is the part of the virus or the bacteria that we're trying to generate the immune response against. In the case of the coronavirus example that I just showed, that would be the spike protein. So we clone or take the gene from the spike protein out of the virus. It's put into what we call an expression vector, and that is a way to express it in a different cell line to make sure that what we're purifying is safe and completely independent from the virus, so you can never possibly get virus back from that cell culture. We make lots of that protein, and we use that as the antigen. The adjuvant is the immunostimulant. So we've been working on immunostimulant research for over 20 years, starting down at a company called Reby Immunochem that was founded in Hamilton, Montana in 1981. So it's actually over 35 years we've been doing adjuvant research. And what that research is we find pieces of the bacteria or a virus or a fungus that your immune system responds to and recognizes as foreign and generates a specific type of immune response. So when we mix the antigen with the right adjuvant, we generate the right kind of immune response to protect against that specific pathogen, which could be different whether you're dealing with a respiratory virus like the coronavirus versus tuberculosis or even influenza or Staph aureus or Pseudomonas or a fungal infection. As you imagine, the immune response generated against each of those different types of pathogens is unique and directed by the different types of immunostimulants or parts of the virus or bacteria that are brought in. So that's the adjuvant component. When you talk about antibodies, the antibody is what your immune system generates to target that virus or the bacteria. So the antibody generated by your immune system will bind the antigen, neutralize that antigen, and prevent the infection.
Great, thank you. That really helps clarify some of the differences between those three words. I feel like sometimes they're a bit interchanged and maybe not always in the right way. So thanks for that great clarification. Dr. Erick Greene, would you like to follow up with the next question?
Sure. First of all, Dr. Evans, thank you so much for the entire team for being part of this today. We know this is just an absolutely crazy time for you and your group right in the middle of I know you guys are just pedal to the metal, flat out, busy right now. So thanks for taking time, and I've certainly learned a lot. Could you talk a little bit about how our immune systems work? And in particular, we hear about our immune systems having memories. And my understanding is that the whole rationale or why vaccines work is we're challenging or giving our immune systems perhaps a taste of what might be to come in a non-dangerous way, but we're somehow getting our immune system prepared with a memory, so to speak, of a future encounter with a real virus. So I was wondering if you could just talk a little bit more about that aspect of our immune systems and immune memory.
No, yeah, absolutely, because that's key to developing a successful vaccine. So when you're exposed to a virus or a bacteria, your body recognizes that. Usually the first thing it recognizes are what they call danger signals or pathogen-associated molecular patterns. And that's what our adjuvants are based on. A virus might come in, it'll recognize the RNA or the DNA from that virus as foreign, and the innate immune system will respond to that in a very specific way. That'll bring in the types of cells needed to try to target that virus or bacteria. When that happens, that virus or bacteria is then taken up by what we call antigen presenting cells. They take up and they break the virus or bacteria into small little pieces and present it on their surface so that other cells of the immune system will recognize it. And when that happens, it's called a cell-mediated immune response. And you get cells of your immune system that many people on the call are probably experts in, or at least have heard of, T cells and B cells. Those cells respond to that infection, and they generate what's called a memory response. In other words, they ramp up and expand, and you can have, a lot of your T cells can be specific to a pathogen during the active part of that infection and eventually clear that pathogen. So rather than just completely going away, when those T cells reduce back down in numbers, some of them become what we call memory T cells and memory B cells. And sometimes they can get down to levels that are even undetectable in the bloodstream. But when you re-encounter that pathogen again in the future, they can rapidly respond to that infection through a combination of cellular responses from those T cells and B cells, or antibody responses, quickly respond to that next invasion of that same pathogen and clear it very quickly. And oftentimes you don't even know you're infected. And I can give one example that's really relevant here as to what we're trying to do in vaccines. And that's, we have an active program on pertussis. And most people hadn't really heard of pertussis other than you got a vaccine from it when you were a child until just in the last 10 years or so. In schools, you start hearing about whooping cough outbreaks. And that's because the vaccine platform changed in the US. We changed from a whole inactivated vaccine, which had a lot of immunostimulants, and it was just a whole bacteria that was inactivated, that was very effective, but had some adverse events to it. And so we went to a safer vaccine, which they call an acellular vaccine, which only has parts of the virus, but took out all that stuff that stimulated the immune system, which was great. It worked very well, but what they found is once people became teenagers, they started losing their immunity and becoming susceptible again to whooping cough. And now you're seeing whooping cough outbreaks in the schools, and people recommend a booster vaccine. And so, for example, we're trying to add back in immunostimulants now into the pertussis vaccine to boost the immune response in a way that gives you lifelong protection again, and trying to boost that memory response.
Great, thank you.
So while we've been discussing some of these questions with you and during your presentation, we've been getting a tidal wave of great questions from our audience. So I'm actually going to transition now to some questions we have from our audience that relate to some of the things we've just been talking about. So Dr. Evans, I'm going to direct this one here to you. Can you address the issue of cytokine storms, or an overdone immune response that has allegedly killed some people with COVID-19?
No, absolutely. So inflammatory responses to either bacterial or viral pathogens is often what ends up killing a person, or just having, significant morbidities. People ended up on a respirator. So the immune response, while it's what protects you and prevents viral infection, if it gets out of control, that inflammation, that cytokine storm, that they call it, cytokine storm, another name for that is sepsis in the case of a bacterial infection. So the immune system is set up in a way of checks and balances. And that is, if it gets out of control, it also has ways to downregulate itself, which some treatments out there, there's a lot of research into how to downregulate the immune system as well. And that's why we have to develop vaccines and immunostimulants that are safe. Safety, it has to be paramount, because if you overstimulate the immune system, it can be just as bad, if not worse, than the viral infection itself. Or you could reprogram your immune system in the wrong way to respond to the virus. And that's why very carefully controlled animal challenge studies, people are always like, well, why are you challenging animals with this virus? That doesn't seem very, very ethical, some people would say. But in this instance, it's required. What if you were to develop a vaccine that stimulated the wrong kind of immune response? And when you got the virus, you were actually worse, because you created the wrong type of inflammatory response. You could get the cytokine storm and you could actually be worse off. And there's examples of that. Antibody-mediated enhancement of disease. It's happened before in clinical trials. So that's why we want to be very safe and careful about the types of immune response we're generating against the virus. In influenza virus, it's usually not the influenza virus that ends up putting people in the hospital. It's the resulting inflammation in your lungs as a result of having the influenza virus. Think about allergies. Allergies is your immune system overreacting to something that maybe it shouldn't.
Great, thank you. We have another question here from our chat box that's a bit more coverage of what's going on with the vaccine worldwide. They're curious about the world's approach to developing a COVID-19 virus vaccine. Would you describe it as a salvo approach? And I'll be honest, I'm not exactly sure what salvo means. So if that means something to you, if you could define that for us, that would be great. Where different groups are trying a variety of different approaches, or is it more like a massive divide and conquer approach, or even maybe something different?
So yeah, I guess I don't know what specifically what's defined and meant by salvo approach either. One thing that is for certain, I have never seen cooperation within the vaccine and research community between companies and federal agencies that's happening now. I've never seen it like this before. So yes, I think there's over 100, 150 or more different groups, probably just in the US, working on vaccine approaches. Multiple vaccines have moved in the clinical trials at a speed that I've never seen before. I know the federal government, NIH, HHS, CDC is working closely with a number of vaccine manufacturers to try to speed up the regulatory approval process, but still do it in a safe way. And so there are many different approaches being tried. Our approach has to do with a recombinant protein combined with an adjuvant. There are a number of our collaborators that are taking a similar approach with their own adjuvants and delivery systems. There's RNA and DNA-based vaccines, different vectors. They're targeting other proteins in the virus. It's really an all hands on deck moment to try to bring people together. There's companies that are donating resources and time, delivery systems and adjuvants to the process. And there's kind of an unprecedented interaction between regulatory authorities and companies to try to ensure this happens quickly.
Great, thank you. So speaking of safety, we have a security question here too. I was hoping you might be able to address for us. If there is viable disease organisms in the labs here locally where we are, on campus or in Missoula or in Hamilton, as you mentioned, what security measures and physical systems are used to prevent accidental release of these organisms that might be infected with the disease for testing and research purposes?
That's a great question. We do not have the coronavirus, the SARS coronavirus 2 in the labs in Missoula at the University of Montana. The challenge studies that we're going to be doing are going to be done at partners who have biosafety level 3 labs up and running, as they do down at Rocky Mountain Labs in Hamilton. Our partners at Mount Sinai, New York, have those facilities available. They all have security procedures in place, both to protect the employees as well as protect others in the community. I've worked at a couple of different biosafety level 3 laboratories over my career, and they all have very stringent protocols in place to ensure, the utmost important, of course, is the employees that work there. And it is every facility I've worked in is safe. I know Rocky Mountain Labs they have a level 3 and a level 4 facility. Security precautions that are in place to even go into the lab will prevent any kind of an accidental release.
Thank you. That feels very reassuring as well. Appreciate that. We have a question here more about COVID, SARS-2, and RNA. We have a comment here in the chat box. Moderna is in phase 1 trials for an RNA type vaccine against COVID, SARS-2. Can you speculate how that would work compared to your approach?
Yeah, no, absolutely. So the RNA and DNA approach towards vaccination of this coronavirus are the ones that have moved the quickest into the clinic because those are the ones that are posed to do that, because they don't have to develop a protein and manufacture that protein and find adjuvants for it. The Moderna approach uses an RNA, an mRNA that's encapsulated in a lipid. And their whole company is based on rapid development of vaccines. They're focused on respiratory viral infections. They've got a similar candidate. I believe it's in phase 2 for an RSV vaccine, which has many similar aspects to it, to the coronavirus vaccine. So their company was based on being able to do this. I think I certainly hope it works. The data that they've generated, both from phase 1 clinical trials and phase 2 clinical trials with other viruses, is very promising. The challenging part is that vaccine platform has never been in a phase 3 study. But I know that they're working very closely with the regulatory agencies to move that through phase 3 as quickly as possible. I believe they're planning to just start their phase 2 study here in June or July, and then their phase 3 study before the end of the year, which is almost unheard of in speed as far as getting a vaccine through the clinic. They've already put in their application for their phase 2 study, even before the phase 1 study is finished. It's exciting new technology. So they basically take the same protein as we're using, the spike protein, but they put it on an RNA and package it in a way that'll get into cells. And then those cells will express that protein from within the cell, and then the immune system will respond to it and generate an immune response against the same protein we're targeting.
Great, thank you. So I'm going to turn it next to Dr. Rosalind Pierre. She has a question she'd like to throw into the pool here as well.
So thank you for joining us this week. Let me begin by saying, correct me if I'm incorrect in this question that I'm about to ask. But the question is, what gives you optimism, given that both SARS or MERS don't appear to have a vaccine? Sort of what gives you optimism that you'll be able to help address this particular coronavirus?
That's a great question, and I would say that we don't have. Of course, hindsight is 20/20, but it's surprising to me, or I guess maybe it isn't surprising to me, that once something is gone, you forget about it. I know there were vaccines for both the original SARS and MERS in development, but once those infections went away, people forgot about them. And if you forget about them, there's no funding for those vaccines. As you saw in my first slide, it might cost $800 million to develop a vaccine through phase 3 clinical trials. Who's going to pay for that if there's no active infection or somebody to sell the vaccine to? And the answer to that is nobody, unless the federal government is going to step up and fund that research and that program to get a vaccine approved. In hindsight, yeah, 2002 we had SARS-CoV. 2012 we had MERS-CoV. We should have known it was coming again, but nobody funded those programs, and so we didn't have vaccines ready to go. In hindsight, we should have, because today's situation would look very, very different. We could have this approach that Moderna's taking. If they had done that with the SARS-CoV from 2002, which hit the same ACE2 receptor, they would probably already have proof of concept through phase 3 and may already have a vaccine that they're administering to people today. So it's more a matter of funding. What gives me promise that we can develop a vaccine is the mutation rate in this virus does not appear to be that of flu. There have been mutations identified in the spike protein. The impact that those mutations will have on the immune system or the ability of the immune system to respond to the virus currently still aren't known. There's a lot of questions out there. If you think about everything that's known right now about this new coronavirus, it's pretty striking to understand that we didn't know about it before December. And to think that we already have multiple vaccine clinical trials ongoing, all that we know about it is really a testament to what the scientific community has been able to do to rally around to make the accomplishments we have. And I've worked on influenza virus for almost 17, 18 years now on a universal vaccine, and it's hard. I mean, we're still working on it. Coronavirus doesn't have the same feel to me. It seems like that the neutralizing antibodies seem to be able to target the virus and clear it in the studies that I've seen. The mutation rate isn't as high. There might have to be multiple vaccines targeting different clades. It might change a little bit over time, but it doesn't seem like an insurmountable vaccine target.
Speaking of timelines, we've got a couple of questions here about the timeline of the development of a potential vaccine as well. For starters, is there any likelihood that a vaccine could be ready by January? And in the news, we've heard a variety of timeframes for the emergence of this vaccine. It could be this year, it could be within 18 months. Can you comment on what's a realistic timeframe? And I realize that we can't expect you to give a definitive answer, that a lot of that is still, it depends. But based on your expert knowledge in your work on this vaccine program, what would you speculate?
Prior to December, I would say no matter what the circumstance was, a new vaccine would at least be 5 to 7 years out. But again, like I said, the cooperation and coordination that's happened at the level of the federal government and companies and universities just in the last 2 to 3 months has really changed my view on that. I really think that the regulatory agencies working together with companies and researchers to rapidly identify and develop a vaccine. Like I said, this Moderna vaccine, they're talking about having it into phase 3 clinical trials before the end of the year. Which I would never believe could have happened, even a year ago. But it looks like that's the direction they're moving, and it looks like they might be able to accomplish it. And they're already scaling up. Then you say, okay, great, you're going to phase 3, clinical trial, but can you make, hundreds of millions of doses of this vaccine? And they're already moving towards that. They're actually moving towards scale up and process to generate the number of doses they need, well before they even have efficacy in phase 2 or phase 3, which is what it's going to take. And a lot of money is being put behind it to make sure that happens. So do I think we're going to have a vaccine at the end of this year in time for, school to start up again in the fall or later this year? I would say no. I mean, I don't think that's realistic. Could we have a vaccine available in 12 months? I think that's plausible. That vaccine might be available for limited sets of people because they still don't have a really good handle on efficacy. The FDA has ability as long as it's safe. That's the main thing. They may not know whether it has 20% or 30% or 90% efficacy, but if they know it's safe, then they can give it to hundreds of thousands of millions of people without risk of making people worse or having adverse events. And they may do that at risk, knowing that it may be only 30% effective, but it's better than what we have today. Let's hope that it's 80 or 90% effective. So I know I didn't really answer your question. I don't know how fast they're going to be able to change the process and speed this up. I'm hopeful that they'll have something maybe available for healthcare workers, maybe people at high risk. Maybe they can target populations where there appears to be an outbreak active and ongoing. And maybe that'll be available, maybe not by this fall season if coronavirus ends up being seasonal, but maybe by the following fall.
Great, thank you. That gives us a little bit, I feel like that information is always changing. It's constantly evolving as we obtain new facts and evidence as the research comes out for vaccines about COVID-19. So in thinking about how we're getting to that, I'm going to turn it back over to Dr. Erick Greene to follow up and ask a question about some challenges maybe in the way with that.
Yeah, sort of related to this, Jay. I can imagine that compared to your normal operations, as you and your team has worked over the past 15 years or so, things are really, really different for you now of how you're operating. And I was wondering if you could just talk a little bit about the challenges of doing your work during the middle of a pandemic. And then I've got a part B after you answer that.
No, absolutely. We really have changed our ways of working within our team. Like I said, we have a group of between 40 and 45 researchers at the University of Montana in our vaccine discovery team. And our number one criteria is employee safety and health. It has to be. Because if we ended up with a coronavirus case in our team, it might shut down all of our research for a 2-week quarantine period. So early on, we were very proactive. We set in place guidelines that have, and I think this should be with all of us in all of our workplaces. If you wake up and we told people, if you wake up and you feel achy or you have a sore throat or anything, don't come into work, stay home. We put in very strong alternative work schedules. People can work from home, all their data analysis, all their report writing, presentations. See, I'm at home right now today. We're allowing people alternative work schedules. We have people working afternoons, evenings, weekends, so we don't have so many people in the lab at one time. We've changed our ways of working. As you might imagine, I'm in a lot of meetings. I always have been. And I've actually found that Zoom can be a very effective way to host meetings where you don't always have to run across campus to a meeting room somewhere else. So I think when this is all said and done and over with, I think some of the ways we host meetings and the way we interact, while there's no alternative for one-on-one interaction with people, I think there's more effective ways to use time as well. And we'll keep some of the practices in place. So we have, as the university has gone through its stage 1 and stage 2 restrictions, we did receive exemptions from the university to keep work because we were working on a coronavirus vaccine. Some of our other vaccine programs around opioid addiction and influenza are also considered essential high priority programs. The opioid crisis is still ongoing in this country. And we're working on an approach to a vaccine that could hopefully help change that. And so there's other essential research programs that our teams are working on. Some of the lower priority, earlier in development vaccine discovery programs we did put on hold. There's no sense in putting those employees at risk working on a vaccine program that isn't essential. And also not put those working on the coronavirus vaccine program at greater risk.
Great, thanks. And my part B is I just heard today on the news that President Trump announced Operation Warp Speed for the development of COVID-19 vaccines. I didn't hear any of the details about it, but given what you've been talking about, timelines and stuff, how do you think this is going to impact vaccine development?
So that's a great point. So the person who was appointed as the head of that is Moncef Slaoui. Moncef Slaoui was the chairman of the vaccines division at GSK for, I believe, about 25 or 27 years, which is the company that I did vaccine discovery and development work for. So I know of him. Like I said, I've met him a couple of times. He is a brilliant scientist, but even better than that, he is used to leading large, complex organizations towards a rapid response. He led efforts at GSK to develop their HPV vaccine program, their pneumococcal vaccine program, a malaria vaccine, and more recently, the Ebola vaccine. When the Ebola outbreak was happening, GSK led some of the efforts to very rapidly develop an Ebola vaccine and get it to phase 1 and phase 2 and phase 3 clinical trials faster than anybody thought possible. Which is why actually I think that he's been assigned this position now. I can't think of someone else that's better suited for trying to figure out how to deal with the regulatory environment and within a company infrastructure and the government and get this done quickly. So I think it's a great choice. He's done it before from inside of industry. So hopefully he can help the federal government do it even better in this case.
Great, thank you. So
beyond that, I don't know anything about what their approach is going to be. My guess is he's going to be at the center of trying to help coordinate the federal government with the private industry and universities and what's going to be needed to make this happen rapidly.
great, thank you. And kind of along those lines, a couple more questions about antibodies and antigens for that safety and security factor here. So this question is going to address one that we got here in the chat box about that. While you hope to develop safe vaccines that do not cause immune systems to overreact in most individuals, some individuals may be more prone to overreactive inflammatory responses. is it possible to pretest and identify individuals who are prone to overreact to antigens and vaccines? And along those lines, there seems to be some variation in the regulation of creating a vaccine with this idea of antibodies. Can you shed some light on that and how that relates?
Yeah, I mean, any vaccine, doesn't matter whether it's the flu vaccine or you know, diphtheria vaccine, you know, DTP vaccine, any vaccine, when you give it to an, you know, in the laboratory, we use inbred mice, which is about as far from people as you can get. Fortunately, people are outbred, not inbred. And that creates a lot of genetic diversity within the population. And yet you might have people who have adverse events or are prone to inflammatory responses. And that's where, you know, the large phase 3, phase 4 safety and efficacy studies that happen with vaccines are so important. But that flies right in the face of rapid vaccine development. So yeah, that is a concern. Is that how rapidly you develop a vaccine versus how safe that vaccine is. When it comes to antibodies, there are documented examples of antibody-enhanced disease happening. They've shown it with the SARS coronavirus, not the SARS coronavirus 2, but the earlier one that came out in 2002. They've also shown it in things like dengue virus, where the wrong type of antibody response, you can actually enhance the infection rather than inhibit the infection. Fortunately, we now have animal models to test for that. So I assume that the FDA will require those types of animal models to be done early in the testing, and they'll also take a very careful look at those when they're moving through phase 2 efficacy studies to ensure that doesn't happen. If someone has some rare autoimmune disease indication or some inflammatory potential that, you know, isn't captured in a patient group in a larger phase 3 clinical trial, you know, that's what phase 4 and post-approval monitoring is for. To make sure you identify those people that might be susceptible to an adverse reaction, identify them, get that information out to clinicians as early as you can to make sure that those patients are informed before they get the vaccine. Did that answer the question?
I believe so. I think so. If it didn't, feel free to add more into for our listeners, feel free to add more into the chat box to further clarify. So we have had a couple of questions come in during that about the idea of a nasal spray. You mentioned that a nasal spray is being tested now as a temporary fix. If the nasal spray testing is successful, how long would it take for a nasal spray to get into the marketplace, or what might you speculate for that?
That's a great question. So the nasal spray that I was talking about was a spray that provides a window, what we call a window of protection, anywhere from 7 to 21 days, where after getting that nasal spray, your immune system was poised and ready to respond to a virus or a bacterial infection via the respiratory tract. Again, that's all done right now, preclinical in animal models. If it works in this study that's ongoing down at Rocky Mountain Labs, there would be a couple of follow-up studies to look at the window of protection and things like that. Once we had that data available, we would then apply to the federal government for monies to move it towards phase 1. And you just heard how rapidly things can move through phase 1, 2, and 3. This would be one of those technologies that's fairly rapid advancement. Because the drug product itself could be manufactured on a mass scale very quickly. We already have a process in place to do that. You know, again, you're talking years, not months, because you still have to go through phase 1, phase 2, phase 3 studies. You know, you're giving an immunostimulant intranasally. So you want to make sure that the inflammatory response is generated after giving that is safe and very well tolerated before you would ever think about moving it beyond phase 2. So we'd have to be careful in the development of that approach. The other approach could be an intranasal vaccine, where you combine the immunostimulant with the spike protein of the virus and use a vaccine that essentially could be effective immediately upon administration, because you could prevent viral infections using the drug-like properties of the innate immunostimulant, and the time it takes your adaptive immune response to develop a memory response, which has a lot of appeal. But again, as a technology that's relatively new, there's no other vaccines out there that do that. So the regulatory path to get there would have to be, carefully waded through to ensure that the vaccine is safe.
Great, thank you. I noticed in your response, there is a lot of talk about different phases for getting to the place where we can have a vaccine available on the market. For myself and for our viewers, can you explain a little bit more about the difference between the different phases of clinical trials and what's needed in order to get to that place that's ready to have it available on the market for people?
No, absolutely. So in general, when people talk about clinical trials, they talk about phase 1, phase 2, phase 3. There's even now a phase 4 clinical trial that people talk about. So phase 1 is the first time in human trial, and that's just simply a safety study. You do increasing doses of whether it's a drug or a vaccine, and you look at safety and tolerability. You want to see, ensure that as you dose escalate up, as you give higher and higher doses of this vaccine to different groups of people, that it's safe and well tolerated. Those vaccines are usually, each dose group might have anywhere from 15 to 20 or 30 patients in it, volunteers. Oftentimes those phase 1 studies don't have any efficacy component at all. Those are not looking for the effect, they're just looking for safety. Sometimes they'll build in some efficacy into a larger phase 1, especially in the case of like here with the coronavirus. My guess is they're also looking at efficacy in the patients they're giving it to. Once you find a safe dose, then you go into phase 2, which are your efficacy trials, where you're looking at duration of the drug response, what is the half-life of the drug in the body, in the system, different dosing regimens, different times, maybe different ways to deliver it, different formulations, different, again, larger groups. Sometimes you can have several hundred patients in these clinical trials. Once you've identified the dosing, the schedule, which specific patient population you're going to target, then you can go to a large pivotal phase 3 study. These can have from hundreds to many, even tens of thousands of patients. And that's where you really want to critically see the endpoint, the efficacy and the safety. You might pick up rare adverse events. We had a question about that earlier. Maybe some patients that maybe shouldn't receive the vaccine or drug. Hopefully your study is powered well enough to pick those up. And finally, after that, you can submit for approval, a BLA or biological license approval from the FDA or the EMEA if you're going into Europe. And then still sometimes the regulators will require phase 4 studies, that is post-approval studies, to follow up either on safety signals that maybe were seen during the phase 3 clinical trial, or just to do larger studies to, again, try to identify other patients that might benefit from the treatment, or again, to look for safety signals or maybe patients that are people that shouldn't be receiving it because of possible adverse outcomes.
Thank you. That was incredibly informative. And I never knew myself what the different phases actually meant. So that's great to know, and helps us understand a better timeline of when a vaccine might be available to us, and be somewhat of an endpoint for something that we're seeing going on right now.
I'd be remiss if I didn't note is that that's the standard process. So once you get through phase 1 and phase 2, you've identified whether a vaccine is safe, and you have a little bit of knowledge about if it's effective or not. You just don't know if it's 30%, a phase 2 clinical trial might show 30% efficacy or 50% or even 90%, but it's on a very limited, 200 people. I don't think there's anything from stopping the FDA from saying it's safe, it showed effectiveness, let's go ahead and launch a product to limited sets of people who are at high risk while the phase 3 study is ongoing. They have
Interesting.
use protocols out there. So the standard process is you move all the way through phase 3 and then you apply for licensure. I haven't heard that they're going to do this, but if you show that it's safe in a phase 1, you show that it has some efficacy in a phase 2, they could actually have a limited product release while the phase 3 is going on. which would greatly shorten the time frame to release of a drug product. Whether the FDA will consider that in the case of this because of the emergency situation and ongoing pandemic, I don't know. I don't have any inside knowledge of that at all, but it is a possibility. And that might be how they're thinking about more rapidly speeding up the release of a vaccine.
Right. This talk of it being available by the end of the year or by January.
It would have to be under that circumstance because the vaccine that I saw, the Moderna vaccine, isn't even going to go into phase 3 trials until the end of the year. So maybe they're considering releasing some limited release of the vaccine after phase 2. I don't know that for sure. I haven't heard that, but it is a possibility.
Great. Thank you so much. This has been a really invigorating episode in our series, and I've learned a lot today. Before I close out, because we've reached our hour time here, are there any last thoughts or comments that our panel would like to share?
Well,
This was a really great session today. I as well learned a lot, and I think that this is going to be great for us to be able to share with our family and friends out there on social media, so that they can take time to listen to this today and really kind of learn a lot and soak it in. So thanks.
yeah, and again, Jay, thanks so much. We know you're just crazy, insane, busy right now. So, you know, you're talking about this is incredibly relevant, and I know I learned a ton. So thank you, and keep up the good work.
No, it's my pleasure. Anytime we can get an opportunity to get in front of people and talk about vaccination, not only on the coronavirus, but on all the vaccines that are available and the importance of those vaccines and how safety is paramount and the development of these is important to the community as a whole.
Great. Thanks. I got to run to another meeting, but well done.
Thank you.
Great. Thank you so much. So I'd like to also take a moment to thank all of our viewers today. We had some really incredible questions, both through Zoom, through Facebook, through our chat box, and we've thoroughly enjoyed this webinar series with all of our panelists. I really want to extend a big thank you to Dr. Rosalind Pierre, Dr. Erick Greene, Dr. Angela Lewis, Brandon Cooper, and today our expert panelist, Dr. Jay Evans. Thank you so much for all of the time and effort that you've put into making this happen over the last 5 weeks. And although today is our last episode in the series, we really hope that our viewers have taken away some useful insights and knowledge about the science behind and living with COVID-19. If in the event we do a future webinar, once more information and more facts and evidence have come to light, we will let all of our viewers know that there is a future episode coming, but for now, we will be taking a bit of a break from our series. As we get deeper into this pandemic, there will undoubtedly be more questions and uncertainty. And as we continue to learn more and experience this new reality, it's important for all of us to search for those real facts, evidence, and truth to stay on a positive trajectory and remain diligent in our safe practices as we navigate through society. Again, I can't thank all of you enough. Thank you to the University of Montana, to the Montana Natural History Center. And as a reminder to everyone, a recording of this webinar and the previous week's webinars will be available on the Montana Natural History Center website. Our co-host Drew will add the link to the chat box if any of you would like to access those recordings. We will also drop that link in the live Facebook chat stream as well. Oh, one last thank as well. Thank you also to Erin Simmons. She was one of our special guests in an earlier episode as well when we were talking about the virus too. So thank you to her and her team as well. Thank you to everyone, all of our panelists, all of our audience, all of our viewers in partnership with this webinar series, The Age of the Coronapocene. Have a safe and fun, socially distanced weekend, everyone, and stay healthy. We hope to see you in the future.